He is getting frustrated by
I think
both.
we'll get started. Thank you for coming to this meeting.
Iran is at its weakest point.
Nice spring weather we're having. Stay cool a little bit longer. Well, cross our fingers. My name is Robert Villa, president of the THS.
Time is running out.
New. This is our mission statement. And we here in the Sonoran Desert are in the traditional homelands of the Tana Atam and the Yaqui people. The region is filled with place names named after amphibians and reptiles. I work at Tumamak Hill, also known as Chamamakdoa, or Horned Lizard Mountain. And across the way in the Catalinas, which are known as Kabadoak to the Atam, which means toad or frog mountain. And that's because of the west side of Push Peak here is looks like a giant toad with its nose up in the air. This is looking, I think, from Shannon Road, the northwest side of town. Okay, we have, this is our most recent issue of Sonoran Herpetologist, which is available for free on our website, TucsonHerpSociety.org. It's a great issue. We continue to produce really great issues. And we have our call is still open for applications to our 2 research grants funds, the Charles H. Lowe Herp Research Fund and the Sonoran Desert Toad Fund. The deadlines for both of these is the 15th of May. One way of supporting our group is through merchandise or donations. This helps us pay our speakers, buy merchandise, fund field trips and other operating costs of our society. We've been around since 1988. I'm not sure this might be, if this is too late or not, but these 2 books here are in the process of being written by various authors, but they are looking for photographs and observations, information that could make it into this book. So for more information, visit this website. We have 2 Gila monster studies in southern Arizona and southwestern New Mexico. Paul Mayer is looking at the genetic diversity of Gila monsters in the Tucson area. If you want to learn more about how to participate, you can email him. And Anthony Pawlicki, who's on our board, is also studying Gila monsters in New Mexico. And if you would like to help him with this or figure out how you can be a part of helping, you can email him at that email address there. And of course, everyone doing these projects are permitted through the state of Arizona. We have 2 volunteer opportunities coming up. This first one is to restore water impoundments that is habitat for aquatic wildlife like leopard frogs and mud turtles. And the contact person is Paul Stone here. And finally, we have amphibian and reptile conservation doing another restoration near Springerville, June 2 through 21. There's a QR code, but if you want more information, see me after the talk. And we are sister members with the newly established State Herp Association of Sonora, and they'll have a virtual and in-person first meeting on May 17th. Before I go any further, Maggie Fusari, our treasurer, is preparing a letter on behalf of the THS addressing proposed change in the definition of the word harm under the Endangered Species Act. This change of definition with the new administration severely weakens the Act's ability to actually protect endangered species. So Maggie will be here later on tonight, but we can provide you with information on where to read more information about this and the letter that we've drafted, which will go out to members or anyone on our contact list tomorrow morning. So I just thought I'd let you know about that. All of our talks are recorded and they go up to our YouTube channel. Next month, we're going to meet 1 week early in May 19th. And this is going to be a very good talk by Randy Babb. Randy is a marvelous photographer. He's a marvelous naturalist and scientist. And he's going to talk to us about the tadpoles and polywogs, the amphibian larvae of Arizona, for the book Amphibians of Arizona. He is actually writing taxonomic keys, which is a very, very difficult and time-consuming process. But he's going to tell us about that. So if this photo is any indication of his photography skills, we're in for a real treat. And so tonight we have Emma Sudbeck, who is a master's student, right, here at the University of Arizona. And she's going to talk to us about bullfrogs. I recently had the pleasure of speaking to Kevin Bonine's herpetology class, and Emma is a TA there. So it's very, very fun to be there, and I appreciate you all inviting me. And without further ado, I'll hand it over to Emma.
Robert.
Yeah.
So I see a lot of friendly faces that I know here today, but for those of you that don't know, my name is Emma Sudbeck. I'm a master's student at the University of Arizona, and I'm in the Arizona Cooperative Fish and Wildlife Research Unit at the university as well. And today I'm going to talk to you about my master's thesis research, which is the population ecology of the invasive American bullfrog in southeastern Arizona.
I think I have the code.
Beautiful. Okay, cool. So our story starts with a frog. This is the American bullfrog. It's a very large banded frog species native to eastern North America. They can grow up to 4 inches long and weigh up to 5 pounds. That's like one of the largest frogs that
5
you can get
pounds?
to. Yeah, 5 pounds.
Wow.
That's insane. It's freaking pancakes out there. But a little bit about their life history is that, first of all, they are habitat generalists. So you can find them in a lot of different types of habitats. I feel like a lot of times we'll think about seeing them in ponds or lakes with like really high density vegetation, like cattails especially. But you can also find them in places like this, where there's just not really that much vegetation. It's just kind of a mud pit, but quite deep. Or you can also find them in some type of lotic systems where it's like not, they're more intermittent pools and not fully flowing. They don't live in like fast water, but you can find them in some lotic systems.
Is that what lotic means? Slow?
Yes, lotic, flotic is what I think. Yes. Pro tip. But yeah, they mostly prefer lentic water, so just like slow moving and rocky water, I feel especially. But typically they do require some type of permanent water to stay in consistently over the years. This is because bullfrog tadpoles typically overwinter. Kind of depends on the region how long they overwinter for, but in like more arid ecosystems like the Southwest, you'll only find them, they'll hatch in like monsoon season, only overwinter once, and then metamorphose next year. That's mostly just because there's like lack of available water, so they probably adapted to this. But in more temperate regions where you have that available permanent water for many years, they can take up to 4 years to metamorphose. So you do need to find them in permanent water. They are pretty aquatic frogs in comparison to the others. Bullfrogs are also a highly mobile species. They are able to move a lot and they move often. In Arizona specifically, where we have kind of more isolated ponds, they've been shown to move up to 10.2 kilometers or about 6 miles within just 1 year. And that was hypothesized just within a few months. In more temperate regions where you have more connected water bodies, they'll move a lot less just because they don't have to move as much, but they still move like about 1 kilometer every year. And this kind of lets them, take over a lot of watersheds and move and disperse really easily. Bullfrogs also have a very high reproductive potential, basically meaning that they're able to reproduce a lot when they do reproduce. So this is a typical bullfrog egg mass. You can see it's just this large, huge sheet that just sits on the surface of the pond. This is about like 2 feet wide. It kind of depends on the bullfrog. It's a very large one. So a typical bullfrog egg mass can have up to 20,000 eggs within its clutch. And bullfrogs can have multiple egg masses with clutches per year. So if you do the math on that, that's a lot of tadpoles every year. Whereas like a native leopard frog species, like the lowland leopard frogs, lay anywhere from like 6,000 to 12,000 eggs per clutch. So that's almost half what bullfrogs do. So bullfrogs are really great at reproducing a lot. So like I said, bullfrogs are native to Eastern North America, and then they've been introduced into Western North America in about the early 1900s. In Arizona specifically, they were introduced in the 1920s for game hunting. A lot of people like to shoot bullfrogs and hunt them, which I think we should bring back, but probably for another day. But they were introduced into a bunch of different states in the West and just really took off from there. They were also unintentionally spread by a lot of states because bullfrog would lay their eggs in like fish hatchery runs for sport fish. And then as you stocked those tanks all across the state with the sport fish, you were also stocking bullfrog types. So that really helped them just take off because they're so good at these other things. They really just outcompeted a lot of, or took over a lot of the sheds. They have also been introduced all over the world as well into a bunch of different countries, kind of for similar reasons. You can see they've been introduced into places like Ecuador, Brazil, Japan, Indonesia, places in Europe like Belgium and Germany, France, all over. So they're kind of a problem in a bunch of different regions. So it's kind of interesting to see all of the different research that comes from these places where there's such different ecosystems or similar ecosystems. But this is a problem that everyone's trying to combat all over the world. So typically, because they're invasive here, you guys probably think of bullfrogs as this evil frog army that's going to take over the world. But I'm going to tell you that bullfrogs are not inherently evil. They're not trying to be evil frogs. They're just really good at being a generalist species and then therefore being invasive. So yeah, but what we do want to say is that they are a conservation concern. They're causing a lot of problems in their invasive range. And this is because of many reasons. The first one being that they spread diseases. So there's a couple fatal amphibian diseases going around, chytrid fungus and then also rotavirus. So these are both diseases that bullfrogs can contract and they're like infected by them, but they aren't very susceptible. So they won't, these aren't fatal to them. But what they'll do is they'll go into some sort of water system and spread those diseases throughout the water. The native amphibians will get affected by that through the water, and they're usually more susceptible to these diseases. So this is actually a picture just from this winter that was taken by another grad student of a chytrid die-off for lowland leopard frogs at the Las Ciénegas National Conservation Area. And this is an event that happens every year now because bullfrogs originally introduced chytrid fungus into this system. So huge concern. Another concern is the lack of predators here in the West for bullfrogs. There's definitely predators that do eat them, like the great blue heron. This is a classic one. And usually when they get older, there's a lot more predators that will eat bullfrogs. But bullfrog eggs and tadpoles are actually unpalatable to most species and predators here in the West. Because they didn't like co-evolve with the bullfrogs like in their native range. So there's a lot of predators in their native range that will keep those bullfrog populations in check, but not here in their invasive range. But more of a concern than the fact that there's a lack of diversity of species that eat bullfrogs is the crazy amount of diversity that bullfrogs eat. So they are voracious predators. They'll eat just about anything that fits into their mouth. And so here are some documented prey in Arizona specifically. So we have the Sonoran mud turtle, desert pupfish, scorpions, tarantulas, red spotted toads and other toads, Mexican garter snakes, invasive crayfish here, Mondrian alligator lizards, many bird species, including the red-winged blackbird, Chiricahua leopard frogs, lowland leopard frogs, a bunch of snakes, including the thread snakes, and even rattlesnakes. There was a western diamondback that was found in the bullfrog center. So crazy amount of things that they eat, and this obviously can be a concern when you have federally endangered species or threatened species in the same system as bullfrogs, and they're preying upon those items. I've talked about how they reproduce a lot and many times per year. So this is a typical kind of haul, I guess you could say, from a stock tank here in Arizona, or just anywhere in their invasive range. These are like hundreds of bullfrog tadpoles within this net that just came from one tank. And so if you think about, if you let those kind of grow up, then you're going to get scenes like this, where the sides of the tank are just absolutely infested with bullfrogs. And if you think about a poor little native leopard frog that's sitting amongst these bullfrogs, they don't really have a great chance of succeeding, especially because they can be preyed upon, they can get disease, etc. So bullfrogs are just able to outcompete most native amphibians in the area in these water systems and take over. So we can't stop bullfrogs from doing these things. We can't stop them from being great generalists and invasive species. Well, actually, there is a study that can stop their reproductive potential, maybe. They're introducing sterile triploid frogs into populations in Belgium, trying to see if that like slows down reproduction. But that's not the strategy that we use here in Arizona. Here in Arizona, the idea is to get rid of bullfrogs at some sort of landscape level to kind of get rid of them so that you're able to reintroduce native species eventually into different places. Just can't stop them forever. So a great example of this that has been done in southern Arizona is helping the Chiricahua leopard frog. So this is a native federally endangered species here in Arizona. And they have been in decline since for many decades, mostly because of chytrid fungus and then also because of bullfrogs, because they just have outcompeted them. So when they were putting together the species action plan for the Chiricahua leopard frog, they determined that one of the main things that's going to help them is to get rid of bullfrogs wherever Chiricahua leopard frogs are found. So they assessed the situation in the early 2000s. And this is a map of the non-urban populations for like areas in southern Arizona. I guess some things to point out here. This is Los Cienegos National Conservation Area. Buenos Aires is over here. Peña Blanca Lake and Rockville Valley. Just get you oriented. So you can see by this that they found that there was a lot of bullfrogs taking over these non-urban areas where there's like forest servicing, whatever. And there's just these tiny little slivers where the Chiricahua leopard frogs were being found. Really sad and obviously concerned when they are federally endangered and trying to bring them back. So a lot of people came together and they were like, okay, we need to get rid of the bullfrogs at some level to be able to use Chiricahua leopard frogs. So you have players like USGS, University of Arizona, Arizona Game and Fish, U.S. Fish and Wildlife Service, BLM, Forest Service. A bunch of different agencies came together, including people you probably know like David Hall, Cecil Schwab, Phil Rosen. They all worked on this frog project. And so now in 2023, this is my favorite map ever to show because it shows hope. This is the distribution of the Chiricahua leopard frogs versus bullfrogs in those non-urban populations. So yeah, you can definitely see that they were successful at reducing the amount of bullfrogs in those areas. They're just down to like very isolated little populations. And then the Chiricahua leopard frogs, there's new populations in some of these places, and they've also spread a lot. So they were able to get rid of enough bullfrogs in big areas so that Chiricahuas could disperse across the landscape, which is awesome. So this is just a great example of why getting rid of bullfrogs is so important. It's not just we're just killing them because they're not native and they're supposed to not be here. It's because they directly it positively impacts native species, like helping get Chiricahua leopard frog back. So there have been a lot of studies about bullfrog ecology across the country and across the globe, but they've definitely been in other regions. So this is a map of the level 3 ecoregions of just the United States. Here's Arizona. It's in the warm desert ecoregion. So a lot of studies have come from more temperate regions in their native range, where they're in a lot of different ecosystems. There's more readily available water, probably more precipitation, and just more like humid environments. There's also been some in their invasive non-native range, but those are also in more temperate regions where it's a lot different in terms of the ecosystem than in southern Arizona. So it's definitely important to think about that because the ecosystems could change the behavior of the bullfrog, and their bullfrog ecology could differ from a lot of these other places where bullfrog ecology has been studied. It's important to look at how that might differ. And we think that might differ because the Southwest is kind of not a unique ecosystem, but it's has aspects that might change bullfrog ecology. Such as, for example, we have like a lack of abundant permanent water here in the Southwest and in arid ecosystems. So there are lakes, there's big reservoirs, permanent water sources, but they're kind of few and far in between. Where and the most prominent water source is probably going to be like cattle stock tanks. So these are basically kind of pits in the ground where ranchers, usually in rangeland, will keep these filled up throughout the year for cattle, usually using like a solar pump or something like that. And amphibians love these. These are kind of the most abundant water source for them. So they're kind of opportunities, use these to breed, etc. Another kind of unique aspect of the Southwest is the fact that we have this monsoon season. So we have this really dry period from about like spring until monsoon season starts about mid-June or July. And so this is really hot and dry. Amphibians aren't moving during this time usually. But during the monsoon season, there's this excess precipitation where a lot of stock tanks are filling up. And so a lot of amphibians wait to breed during this time. And it's thought that bullfrogs will wait to disperse across the landscape during this time because there's more available water and there's just going to be less risk of desiccation as the bullfrogs are moving across the landscape. So there's a lot of people that know a lot about bullfrog ecology in southern Arizona. But that knowledge just hasn't been included in literature. So it hasn't been able to be shared amongst other arid ecosystems in the United States or even in like other countries across the world. So what my study hopes to do is to fill those knowledge gaps in that published research to make sure that we can share this information about their ecology to other places that might be wanting to start some sort of bullfrog eradication project at a landscape level or whatnot, or inspire them to do studies similar to mine, because there's evidence of some sort of difference between their ecology in the more temperate regions versus like arid ecosystems. So to talk about my study, I'm going to talk about 3 different study goals today, and we're going to focus on the first one first, and this is to determine the dispersal distances and the influence of landscape features like topography or drainages on bullfrog dispersal. And the goal for this is to basically advise the design of buffer zones for bullfrog eradication and control. I'll talk about what buffer zones are. So my study takes place in the San Rafael Valley. This is a valley that's about 2.5 hours southeast of Tucson, really close to the border. And this area gives us kind of an interesting landscape to look at in terms of topography features that can influence bullfrog dispersal. It is a valley, so in the center, you have more flat habitat that's grassland. And then as you move up towards the mountains that are flanking it on either side, it becomes a lot more hilly. We get into more woodland oak habitat and more drainages, whatever. So there's a lot that we can look at to see what aids bullfrogs in moving across the landscape or what might act as barriers to dispersal. So the San Rafael Valley is home to all kinds of critters, furry and smooth. But of course, I'm going to talk to you about the not furry ones because this is the Tucson Herpetological Society. So just to highlight some of the home species, we have the Mexican garter snakes, king snakes, including the desert king snake. We have little tantillas, black-headed, this is the Chihuahuan black-headed. We have garter snakes like the checkered garter snake and black-necked. We have Sonoran tiger salamanders, canyon tree frogs, Arizona tree frogs, Sonoran desert toads, red spotted toads, and Mexican spade toads. So all kinds of species have the San Rafael Valley as their home. And this just kind of gives us like, I guess, a passion for trying to preserve this area because it is home to such important creatures, including many mammals and many birds that also run around this area. So working to kind of combat the bullfrogs in this area definitely would make an impact on the species. So, like I mentioned, the main source of water in a lot of these places are cattle stock tanks, and that the San Rafael Valley is kind of the same. So, but this also we're able to look at a lot of pond level features in the San Rafael Valley that might also act or like influence how bullfrogs are dispersing or moving across the landscape. Because you get really lush vegetation like this, where it just looks like frog heaven with all of this vegetation, really nice water. But you can also get tanks that look like this, where it's just a literal mud pit in the ground with cow poop pond. But you'll find bullfrogs in both of these, so it's kind of provides an interesting thing for us to look at, whether bullfrogs are using certain habitats during dispersal or just during overwintering, whatever. There's a lot of variety there. So before I talk about what we did this summer, I want to talk about why dispersal distance is an important thing to measure and study. So let's put you in the place of like a wildlife manager or a land manager of sorts, and you have this big landscape that has populations of bullfrogs on it. So each little bullfrog is its own like population. Let's say that you really want to get rid of some of these so that you can eventually reintroduce Chiricahua leopard frogs. So you do that, but you're only able to get it down to just 4 tanks. So there's still a few populations of bullfrogs. Maybe because you didn't have the time or the resources to get rid of these, to get rid of all of them. Or maybe these are on private land where the private landowners just really love bullfrogs. But nonetheless, you can't get rid of them. So, but eventually you reintroduce your Chiricahua leopard frogs. So you have a little population of them on that west or east side. But you kind of have this area in between where you don't really want them to mix, because bullfrogs are going to create that direct threat on the Chiricahua leopard frogs. So you want to make sure that you're kind of monitoring this in between part to make sure that the bullfrogs aren't going to reinvade where the Chiricahua leopard frogs are. So this is your buffer zone, and the width of this buffer zone can be determined by that maximum dispersal distance of bullfrogs. So once you know that bullfrogs can move this far, you want to make sure that your buffer zone encompasses that, and then as you're monitoring every single year, you're making sure that if there are any bullfrogs that show up, you can get rid of them before they fully disperse and reinvade the population of the Chiricahua leopard frogs. So that's why it's important. But to get on to what we actually did. So in 2024, our field season went from about May until mid-September, so over a 5-month period. And it's kind of important to note that there's 2 seasons at play here. So we have our pre-monsoon summer from May until about mid-June when the monsoon season starts. And this is basically where it's really hot, really dry. There's no precipitation. So it's hypothesized that frogs aren't moving during this time. And then for the later half of the summer, we have the monsoon. So that's when that excess precipitation comes. Ponds are filling up, bullfrogs are moving across the landscape. So during that dry season, we wanted to first identify source populations. And these source populations are places where there's active reproduction. So like tadpoles, signs of egg masses, or we were looking for places with just a high abundance of bullfrogs, anywhere from 10 to over 300 individuals. The idea is that, you know, when you have a really high density population, the individuals are going to be more likely to move out from those populations so that we can get them as they're dispersing across the landscape. So once we identified those source populations, we then marked all of the frogs at each of those 4 source populations before they were going to start moving across the landscape. And then once the monsoon season starts, we would survey the tanks or the ponds surrounding those source populations to be able to hopefully recapture the frogs that we marked at those source populations as they're moving across the landscape. So this is a map, kind of crazy map, of all of the tank locations in the San Rafael Valley. But the most important thing to look at right now is the fact that we identified 4 source populations. So again, places where there was active reproduction, tadpoles, egg masses, or just a high abundance of bullfrogs. Conveniently, they were all right next to each other, so that made my study site really nice. But yeah, so those were our 4 source populations. So the majority of the summer was definitely spent grabbing frogs and capturing them. We did this in 3 different ways. The first way was with these collapsible minnow traps. We would bait these using cat food, specifically chicken liver. And the bullfrogs really like these. We would usually get juveniles and small adults this way. Also tadpoles, except we didn't need tadpoles, but if you're looking to get tadpoles, it's a good way. So we captured them this way, but again, we didn't get many big adults. So my idea with these traps was to get more big adults this way. So we had these turtle traps, where the idea is that the bullfrogs would come up on those ramps and then be dropped down into the trap. And these kind of like float in the middle of the pond. These didn't work. We did not get a single individual with these, but I'm gonna try again this way. Sorry.
Just for locating.
Yep, and then, but definitely the most efficient and effective method of capturing bullfrogs was hand capturing. So we would do this using a spotlighting technique where we have a really bright flashlight and you can kind of stun the frogs and then you're able to get really close to them enough to just grab them. And you get handfuls of frogs like this in some of the tanks, so definitely very effective. So once we captured frogs and had them in hand, we marked them using toe clips. So each source population had its own unique toe clip combination. And this toe clip combination was 1 4-foot and then also 1 9-foot. So each source again had their own unique toe clip. And for this, we only clipped 1 toe per appendage, and we didn't try to clip any important toes to try and not inhibit any. So yeah, that's how we marked them. And then, so these are the numbers. So we did a total of 87 night surveys over the course of 15 weeks. We marked a total of 323 frogs at those 4 source populations. We had 6 flooded waders, which is kind of a large number, but people are really committed to grabbing the frogs, so I'm not gonna complain. And just 1 flat tire, which actually under the whole field season, not that bad. But it was a pretty bad one. That was a lot of
Wow.
impressive. But anyway, back to my study. So this is a pie chart showing, so it's important to note that the amount of frogs that we marked at each source population was super uneven. So we have 4 source populations, like I said, and most of the frogs that we marked came from 1 tank. And this tank was jackpot tank. We had 244 individuals out of the 323 come from this tank alone. And this tank has probably over 500 individuals at it. So we just barely scratched the surface. We spent a lot of time at this tank trying to get as many as possible, but yeah, we still weren't getting a lot of like recaptures when we were coming back. So we tried our best, but we still didn't get a lot. But at the other 3 source populations, they had much lower numbers. So they had anywhere like, 25 to 28 individuals, maybe a little bit more, but we definitely marked the most, or like the majority of the frogs at those source populations because it was a little bit easier to get to those numbers. But keep this in mind. So here's this map again with all the tank locations, source populations. So over the monsoon season, we surveyed 41 total sites, and this was the sites that we actually got to survey. We didn't get to the other ones due to time, resources, etc. And of those, we found about 14 that had bullfrogs in them during the monsoon season. And so this was, either dispersal during the monsoon season or from the source populations or from other populations. We're not super sure. But we did get actual results for movements from those to and from those source populations. So these are the results. So we saw movements to and from 3 out of 4 of the source populations. And then we saw movements to an ephemeral tank, which was a tank that we surveyed prior to the monsoon season. Didn't have water, no frogs. And then during the monsoon season, it filled up with water and had frogs. So frogs were definitely dispersing to this tank. So it makes sense. So our first movement was found from jackpot tanks, surprise, surprise. That was the tank that had over 500 individuals. And we saw movement from there down to this other source population, pasture 9 tank. And this was 3 juveniles that we found that moved 1.11 kilometers. And that was about the size that they were. Then we had our second movement, which was our longest. This was again from jackpot tanks, surprise, surprise. To max tank, that ephemeral tank. And this was 1 juvenile that moved 2.28 kilometers, or a little bit over a mile. And that's about the size that he was, or that is the size that he was. And then lastly, we found movement from that top source population, big boy tank, down to max tank, that ephemeral tank. And this was actually one adult female that moved 1.15 kilometers in total. And in comparison to the size of the frogs that we usually get seen dispersed, this lady was a lot larger. This is a big lady with a big titty. So, some interesting things to note in terms of landscape features that bullfrogs might have moved to disperse. Our longest movement, this one, the 2.28 kilometers, those two tanks are connected by a drainage. So most likely that juvenile moved up through that drainage because that would be certainly a great cue to look for if you're trying to find water somewhere else. But these other two movements, these shorter ones, if you kind of look closely at this map, see that they're moving over some crazy hilly topography. There's big ridges in between these two tanks. If they're moving that straight line topographic distance, hills, you know, everything like that. So if they were moving that straight line distance, they were moving over some crazy topography. So we were like, how do we actually figure out what the paths they're moving are? Because are they actually moving down this drainage, or are maybe they looking for some flatter land to move across and then down a different drainage, similar to the other one, so that they're actually moving a larger distance than what we actually found. So we wanted to try and determine their actual path using telemetry. So I made these little fanny packs for the frogs. There is 28 total of them that we put out. So on the left side, these are VHF telemetry transmitters. And so that basically they emit a signal from those little antennas that then we're able to pick up using like a radio. So we're able to kind of get more like track movements more specifically from these frogs. And then the ones on the right had a more fancy transmitter where it was a combination of that VHF, and then they also had a GPS on that. So it kind of works like your phone where it's sending out the signal to a satellite space or whatever, and tracking individual movements like per hour. So only 4 of those got the fancy special ones. Most of them had these other VHF transmitters. So we sent them on their way and we told them to go do big things, give me good data, and hope that they listened. And we tracked them 2 to 3 times per week for about 2.5 months. So we would go out and we would check to see if they're in the ponds or not. So we had to, or the GPS, the fancy ones, those only lasted about 2 weeks, unfortunately. So we had to recover those a little bit faster just because they were giving us hourly fixes. So they died really fast. But the VHF transmitters were alive for the whole 2.5 months. I want to note, when we were going to look for these frogs, we weren't tracking the individuals to their individual place in the pond, because that's not what we were looking for. Just like if they're moving or using certain habitats in the pond, we would just go to the tank and make sure they were still in there, or see if they had moved out from the tank, and then we were tracking from there. So unfortunately, when we went to recover them, we saw that over half of them fell off the frogs. So over half of them are just stuck in this really dense vegetation and like deep into the mud. So we were digging these out for many hours. And we also kind of hypothesized that the other half of those were also fell off the frogs, but they were just too deep to recover. So that was kind of a bummer. It was really cool to be able to learn VHF telemetry this way and to learn these skills that I'll hopefully use in the future. But we didn't get any data from this, unfortunately, except I guess that it makes sense why there's no bullfrog studies on telemetry. So I'm joining the club. So yeah, in summary, we surveyed a total of 41 sites over the course of the summer. We documented movements from 5 out of those 323 total individuals that we marked. And those were the 1 adult female, which was quite large, and then 4 juveniles that we saw moving. These movements range from 1.11 kilometers to 2.8 kilometers, and the median was about 1.15. Telemetry doesn't work, but I guess I do have some ideas of how it could work better if it was specific. I'm not going to be doing that. All right, so next we're going to talk about the reproductive potential of bullfrogs. So now that we've covered that first study goal, we're going to move on to the second one, which is to determine the size at which female bullfrogs become reproductively mature. And hopefully this will advise the prioritization of certain site classes during that first year of eradication. So why is studying reproductive maturity important? Let's take this big lady, for example. So she probably, if she were to lay an egg mass, she would lay a large egg mass with a lot of individuals. And then therefore, if she moved to a new pond, laid an egg mass, there's then tens of thousands of individuals that you're gonna have to worry about during an eradication project. But if you were able to unfortunately take her out before she lays those egg masses, then you would have to deal with a lot less. So stopping reproduction is one of the first steps of eradication so that you don't have to worry about all those tadpoles and metamorphs the next year. So this part of my study takes place at the Babakamari Ranch. This is a river system that's kind of like an hour north of San Rafael, like an hour and change southeast of Tucson. This is a really lovely riparian system in southern Arizona. It's one of the last ones that's really good. You can see it's very lush in terms of vegetation. It's not fully flowing at all times of the year, but it does have water at all times of the year. So this place is home to a bunch of different creatures, lots of mammals, birds, and especially bullfrogs. This is home to a huge population of bullfrogs, has been for a long time. And this is a private ranch actually. So the people that own this ranch, they're really interested in conservation, helping out or trying to renovate their system to make sure that it's beneficial for wildlife, which is great. And so because of this, it was kind of the next prioritization in terms of sites for bullfrog eradication with that frog project. So you can see here, this is the Babakamari. It's this little red river system that's circled. And then we have Las Ciénegas up here, Dalton Battelle down here. And then we have these buffer zones in between. So the Babakamari was a high priority site to start because it serves as a source population of bullfrogs that could disperse into that buffer zone for the Las Ciénegas National Conservation Area, and or they could move into the Appleton Battelle Research Ranch, where there is a population of Chiricahua leopard frogs. So in 2024, they started this project. This is Chris Pruitt. He is the project manager for the Babakamari Frog Project. And so basically, what they do is they go out every week and they shoot the bullfrogs using a .22 rifle. And they kind of use the same spotlighting technique that we use to capture the bullfrogs. They shoot them instead. So they're going out every week doing this, and we decided that this would be a really great opportunity to collect these carcasses and get some more information from them. So my wonderful technician Morgan, she was kind of the head of this project, which was awesome. This took a lot of work on top of all the dispersal project stuff as well. So she would go out every week, follow these guys around, and then scoop up any of the carcasses that could be recovered from this project. And then once we had them, we would take them back to the university and dissect them. So dissections, we were looking for two different things when we were doing these dissections. The first thing was the reproductive maturity. So we were looking at the ovarian stage of females. And then we were also looking at their stomach contents. Over the course of 4 months, we were able to dissect 363 bullfrogs. So a lot of great data with this. So now I'm going to show you some bullfrog dissection photos, which are a little bit graphic, so I have a little warning. There's no blood or anything, it's just kind of like gross bullfrog guts. So just a warning. So like I said, we use ovarian stage to determine the reproductive maturity of the bullfrogs, female bullfrogs. So this is a stage 1 individual. Here's the ovary, I've circled here. You can see that it's very orange, it's kind of small. You don't really see eggs within this ovary. So this is a very reproductively immature individual. You'll see juveniles just coming into adulthood, I guess, have this. Then you have a stage 2 individual where you see that change in coloration of the ovaries. The eggs are starting to kind of develop. See down here. And then stage 3 is where you it starts to look a little bit more like actual egg mass, like you've seen. You get more developed black eggs, but it's kind of more half and half with those yellow undeveloped eggs. And at the end of stage 3 is when individuals are technically reproductively mature, but that's really hard to tell visually. So We just identified stage 4 individuals as the reproductively mature ones. So then this is a stage 4 individual. The majority of those eggs are a lot larger, they're black, they're definitely developed. And so this individual will lay an egg mass within the next few months. So this is what we found in terms of trends. This is the data that came out of it. So on the y-axis, you can see the snout vent length of a frog, or basically the length of them. And then the x-axis is those ovarian stages from 0 to 4. I didn't talk about stage 0 frogs, but those are basically juveniles that you can see ovaries in, or they just had like fat bodies that were just beginning to be ovaries, super reproductively immature. You can see those are mostly juveniles. So see here that our smallest size at which we saw reproductive maturity, or stage 4 frog, was 120, a female that was 123 millimeters long, or about 12.3 centimeters, or 4.8 inches if you take the metric system. So some interesting things to look at here is the fact that there's a huge overlap of the sizes of the frogs versus their reproductive maturity. So any individual from about 123 up to this like 160, 170, they could be reproductively immature at a stage 2, or they could be reproductively mature at a stage 4. So it's hard to tell just from looking at them, but if you see a frog within this size class, you should probably take it out and decrease that risk of reproduction. So that's it for the reproductive maturity part. In summary, we saw a positive relationship between body size and the stage of reproductive maturity. We saw a huge overlap of the sizes of the females from stages 2 to 4. And the smallest size at which we saw a female being reproductively mature was 123 millimeters, or 4.8 inches. Okay, and then lastly, our last study goal is to identify the prey items in the stomach contents of bullfrogs that we dissected. So these came from the same location. So like I said, bullfrogs are voracious predators. They'll eat just about anything that fits into their mouth. Each other is no exception. They do show a lot of cannibalism usually. And this was a picture from this year, actually. We put them all in a laundry basket when we're done processing them. And I guess these guys had beef with each other and decided to eat one another. We did in fact pry the frog out of its mouth because he was clipped, and that's data. So that was a good adventure. But anyway, okay, so what we found. On the y-axis, we have the number of bullfrogs, and this is out of that total of 363 individuals. And then on the x-axis will be the groups of species that we found. We did pretty general groups. We didn't do like to a certain class or order or anything. We just kind of looked at it and said what it was. So the first item that we saw was definitely crayfish. The Baba Kamari is full of a lot of crayfish, so this makes sense. We have other arthropods because crayfish are an arthropod, but these are like more insects, including spiders and moths and bees, all kinds of wasps, beetles, things like that. We saw plants and debris, which are kind of similar, just like plant material. Bullfrogs don't tend to go towards this, but I think it's just more like bycatch when they're going for something else. They just latch on plants. So we didn't find like huge amounts of this in their stomachs. We did see evidence of them preying upon fish, worms, invertebrates, mammals. We got some mammal fur, and we did get a couple birds. This was feathers, a really digested bird. Can't tell you what it was because it was so digested, unfortunately. And then a reptile. So the most prominent prey item that we found in the Baba Kamari frogs was definitely crayfish. Over half of the frogs had crayfish within their stomach. This is a really prominent prey item at the Baba Kamari. So if you look at the Baba Kamari River, you're going to see bullfrogs and crayfish. So it kind of makes sense. The second most prominent prey item was the other arthropods, so insects and spiders. We saw a lot of those. So majority of frogs had either of these two prey items within their stomachs. Second, or third to that would be plants and debris. We saw a lot of prominence of that, but at low amounts. And then after that, we kind of saw a very low count of all of those other categories. So only a few with fish, a few with worms, and one with mammal, one with bird, one with reptile. So some highlights, I guess. So this was a crayfish that we found in a bullfrog. This was a huge crayfish, the largest one we found. So the crayfish measured from its snout down to the end of its tail was 110 millimeters. And this bullfrog was 150 millimeters. So there's only a difference of like 40 millimeters between the size of this frog and the crayfish. So I can't even imagine how long he's gonna sit there digesting this thing. And then this was the reptile that we found. We saw a checkered garter snake within the stomach. This was about 220 millimeters long, or kind of like juvenile. It was kind of interesting that this was the only reptile that we saw, but there aren't that many reptile species that have been seen at the Baba Kamari. So garter snake would be the most likely one. Then some honorable mentions, I guess. We saw quite a few bullfrogs with tarantula wasps in their stomach, which I found kind of crazy because they're stingers, you know. We've had quite a few of those, which is wild. And then these were the fish that we found within one of the stomachs. Yeah, I think there's some kind of centrarchid, like sunfish and bass, and then a mosquito fish. We saw a lot of mosquito fish. I've sent these pictures to like all of my fish friends, and nobody can really identify them just on the picture. So I'm just going with some sort of centrarchid. I didn't save them, so that makes it. I know you were waiting. Yeah.
Just so yellow.
A testes. And this is an overdose.
Okay.
Let's put that. So yeah, those were the most common or most interesting prey items that we found. So when we look at prevalence of prey by body size, we found that smaller frogs were more likely to have other arthropods within their diet. So like smaller things like insects and spiders and such. And then larger frogs were definitely more likely to have crayfish within their stomach. So this kind of makes sense. The larger frogs are going to go for the larger prey that's more hard to digest and eat, whereas the smaller frogs are going to go for more easy prey items like insects and stuff. And then we also did test the relationship between vertebrates, like we lumped the mammals, the birds, and the reptiles together. And we found a positive relationship, but it was a weak relationship between, so like larger frogs were more likely to eat vertebrate prey. But we had a really small sample size, right? We only had like 4 individuals that had vertebrates. So really weak relationship, but I think it would be more prominent in other systems, and it makes sense as well. So in summary, what we found was that the Baba Kamari frogs primarily prey upon arthropods, specifically invasive crayfish in the system. Smaller frogs tend to go for insects and spiders, and then larger frogs tend to eat larger prey like big crayfish and then vertebrates as well. We did not have, or we had very few reptile and amphibian prey, which is kind of an interesting thing if you think about it. Usually bullfrogs go for a lot of those items. But the records kind of reflect the available prey sources at the Baba Kamari, so this makes sense. There's also been some other studies that have been published about the fact that if there's bullfrogs and crayfish in a system together, bullfrogs are going to go and focus primarily on those invasive crayfish and kind of ignore the other reptiles and amphibians that are less commonly interacted with bullfrogs. So our study kind of also makes or like adds to that. So yeah. So this was just one field season. This was just 2024. So we have a whole nother field season this year to do all of these things all over again and collect even more data. So along with doing everything that we're doing this year, we're also going to be pit tagging individuals this year. So this will hopefully give us more data on like individual movement from pond to pond. If they're moving from multiple ponds, we can see that versus before we were just able to look at whether they got from point A to point B, didn't know their path. So hopefully we'll be able to look at that. We're also doing dissections on frogs collected from the San Rafael Valley. So we're going to add a lot of data for the reproductive maturity stuff and then also the diet as well for those frogs. Similar to that, we're going to be incorporating data that was collected by David Hall and a bunch of the other frog project crew from the past few years, about a decade. They collected a bunch of also reproductive maturity and diet information from a bunch of ponds around southern Arizona. So the scope of this study is going to eventually be much larger than just the Baba Kamari River, which is awesome. And I hope you all get my eyesight reference. All right, and so lastly, I just want to acknowledge everybody who has helped me through this field season and the chaos of it. I've been supported by a lot of really awesome people, and I couldn't have done it without any of them. Special shout out to my technicians, Morgan, Riley, and Jeremiah. I really could not have collected the amount of data that I did without them. Similarly, all of the volunteers that came and helped. When you're getting huge handfuls of frogs like that, you definitely need a lot of hands for help. So yeah, thank you to everyone who has helped me and supported me. Yeah, so much info.
Hey, I've never looked at fish eggs under a microscope, but can you do it? Like whenever you're looking at,
You mean frog eggs?
what did I say?
Oh,
Oh, that's my girl. Sorry. When you're looking at frog eggs, can you tell the difference like in between stages by looking at them under a microscope or no?
Yeah.
Okay.
Yes, yeah, you can. Or like within the dissection.
Yeah, do
That
you guys
is-
need to do that?
Oh, we didn't do that, no, but that would be maybe good. You don't really see like they're not fertilized, so you didn't see like development of like a tadpole or anything within them, so I'm not sure if you could like look at a microscope and see the reproductive maturity just based on that. But perhaps it'd probably be easier to look at like that ratio of underdeveloped versus developed eggs, so it's a good idea.
Staying on the reproductive maturity stuff, did you look at or do you have plans to potentially look at the effective season on also that the level of reproductive maturity, like as a, you know, combined effect
Yeah,
with
let's-
size?
Yes, definitely, yeah, so that's kind of what I'm excited about, the fact that we're gonna do dissections on the San Rafael Valley frogs, because that's gonna be in the fall, so. Most of the ones that we got from the Bobby Kumari are in the spring, and then those ones will be in the fall, so we'll definitely be able to look at that seasonality, which would be super cool, yes.
Has there been any evidence that these guys can crossbreed with our native frogs?
Yeah, I don't think so, I have not seen any evidence of that, thankfully.
Yeah.
That's a really good talk. Thanks for speaking here. I have so many questions, but is there anything about a place that makes it more unfavorable for a bullfrog?
Good question. I feel like what I've learned is not really, because I would think the desert in southern Arizona would be extremely unfavorable for them, just because there's not that much like permanent water, but they seem to overcome that and
Yeah.
they just move further distances to find the water. So I feel like not really, but I guess like higher elevations, they really don't like cold water. So you don't ever find them. Like, mountain streams or anything like that, which is good.
Have you looked at whether they're vectoring parasites?
No, I haven't. I don't think so. I mean, I think, yeah, coronavirus, that's.
Yeah. There's a guy named Richard Shine in Australia who, maybe you've read this news item, but using genetically modified cane toads, invasive cane toads, to produce tadpoles that never leave the larval stage. With the idea being that they eat tadpoles that don't.
Right, yeah. Interesting. Do you think we should do that for bullfrogs?
I don't actually.
They stay larval stage way too long.
Yeah, life just always finds a way.
Truly, truly. Yes.
So thanks, Emma, that was great. So what are some effective, or what have people found recently is the most effective way to eradicate bullfrogs? And is there any method that's
Yeah.
proven?
I think the best method definitely is shooting them. That seems to be the most efficient because you can get a lot all at once. And then you don't really affect the other aquatic species in the river. You can't just like, poison. The poisoning the whole stream doesn't work very well for bullfrogs. Then you're also affecting the other aquatic species in there. Yeah, I think that's definitely the most effective way. I think they do like seine nets too and stuff for tadpoles. Works really well for getting a lot of individuals. Yeah. Kind of scooping them up, I guess, also euthanizing them would probably be similarly good, but sometimes bullfrogs are hard to get close to, so being able to be far away from
Is there anything they will eat?
that. Well, I feel like not really, I guess.
I'll send you a photo of one that's that caught a bat.
Oh,
yeah.
And birds, good place. Yeah.
Yeah.
Really want to see one catch a bird.
Yeah.
That's on my bucket
Yeah.
list. And then safe bird, obviously.
There's one last thing I want to say, which that really stuck out to me at the beginning of your talk, is that I think it's so easy for us to villainize invasive species, right? And the very word invasive is kind of really has a lot of baggage that associated with it. And I think it's really great that you emphasize that these animals are just doing what they've evolved to do, right? And that framed it in a way of is the invasive species, is it a good neighbor? Right, as a metric of understanding how important it is as a threat, right? And so you're asking the bullfrog question against, the health of our native species, and not just it's not from here, so we should just kill it, right? So I really appreciate that, and it reflects something that I heard from an indigenous biologist saying, when we talk about invasive species or introduced species and the threats that they pose, a good way of assessing the issue or the problem is, are they good neighbors? And that's really how you frame the question. Are they good neighbors with charcoal leopard frogs and all of our other native species? So I just wanted to say how much I appreciate that. And emphasizing, look, they're not inherently bad animals, right? But we have to do something because we know that there's a negative impact.
I feel like it's a huge topic nowadays, of like, are invasive species, you're supposed to just take out all of them, or they're technically just really good at living in a certain area, so at what point do you just kind of let them exist? And I think being a good neighbor is definitely a good way to think about that, because if they can coexist, then you should just be able to lead them at some level, but
Yeah.
they're definitely gonna be a threat to biodiversity, and that's but you gotta have a problem.
Kind of in line with things being good neighbors, it's really interesting seeing the prevalence of crayfish in their diet, both because that's native prey for them and also because they're providing a bit of a service, right?
Sure.
Do you know if any conversations are happening about what it will mean for crayfish populations as we eliminate bullfrogs from watersheds?
A really good question. I think that is definitely starting the conversation, because we're kind of realizing that in these systems, a lot of times it's crayfish and bullfrogs. And if the bullfrogs are kind of controlling that invasive crayfish population, then you get rid of them in the system, what's going to happen to the crayfish, right? And then how do you combat getting rid of the crayfish as well? I feel like that's the next step that I've heard a lot of people talking about. I don't know exactly how they're going to do that. It seems hard, but yeah, it's definitely a conversation now.
And crayfish are definitely harder to deal with.
Yes, yeah. It's gonna be an interesting battle,
Yeah.
that's for sure. Yes.
Did you get any good bycatch?
Oh, like are in the traps? Not really. We didn't get, we got a lot of salamanders, like salamander larvae. So it's a cool bycatch. But a lot of times there's not really fish in those tanks at all. So we never got any fish. Didn't get any garter snakes, bullfrogs, salamanders.
Oh, is the female suit doing captive being bullfrogs? Leopard?
Yes.
Oh, is that going well?
I think so, yeah, I think they do a really good job at breeding the two recall leopard frogs, and that's where they get the ones that they've been reintroducing some of those populations in that map. They get them from the Phoenix Zoo and reintroduce them. Yeah, so I think it's going very well.
I know, it sounds like you're switching tactics since the little fanny packs didn't work out. Super great. As a fanny pack wearer myself, I appreciated you, calling it like that. So I know in the southeast, like there are folks who have and are currently still doing like some tracking on reined frogs still with telemetry stuff. So if you were interested, I could probably get you some contact information. So like, I don't know, maybe the gopher frogs were just like, we like our fanny packs. We don't want to get rid of them. Or like, I don't know if they've got like some other methodology for that. So that's part one. And then two, since you're gonna do the pit tag stuff, is that just so, you know, like how is that? How are you going to be doing that? I guess I'm just curious about like is it just gonna be like setting up an array around your source population tanks and then just seeing like when they cross over that, or how is it just gonna be like scanning ones that you catch it like the other ones?
Yeah.
I guess that's the question.
So it would be really cool to have a pit tag array. I feel like that would be the ultimate goal, but I don't have the resources nor the money for that. Second year of this study. So we are just going to like, once we capture them and have them in hand, we'll just kind of like scan them for the pit tags. So it'll just be another way to identify which population they came from. And then, because before we were able to like mark them again, to determine whether they were moving again after that. Because there's just like too many total combinations in play there. But so I think that'll really help with getting actual movements like from pond to pond. And then we could even do like survival stuff if we really wanted to. Yeah, it's just extra data, I guess, yeah.
As fluorescent ink, was that ever a possibility, or is it expensive, or?
No, it's not, but we found that the bullfrogs seem really thick.
Oh.
So it's like really hard
Oh,
to be able to
it's
see.
opaque,
Yes.
okay,
Yeah,
so it doesn't come through.
not really, yeah. And we've tried like VIE tags too, like little identification codes. Those just like disappeared into their legs, so.
Right. Like
Yeah.
like a punch, like a,
It was like an injection, yeah, like a little.
Yeah.
Goes into their skin with a little tag. It's too thick skin, but we did think about that, yeah,
we really have figured out how to evade us.
for sure. Yeah, they're really smart frogs, really. Little brains.
Well, thank you so much.
Thank you for having me. This was fun.
Yeah, Maggie has something to announce about the letter we're writing.
My apologies, especially to you. This topic is fascinating to me. I had a commitment I could not ignore, but my students and I on the Santa Cruz campus dealt with bullfrogs. They are not friends of the California redwood.
Very true. Not good neighbors.
So this is a very interesting topic, but not anywhere near as uplifting, and I apologize for that. Tomorrow morning, around 5 o'clock, your email boxes will receive an email from the Tucson Herp Society with an explanation of stage 1 of undermining the Endangered Species Act. Some of you may have already heard about it. If you haven't, you should. And I've got some links in the email if you want to explore it in more depth. This particular thing is based on something published in the Federal Register. And probably you all know that in the Endangered Species Act, it forbids take and defines take, and one of the words it uses to define take is harm. The publication in the Federal Register redefines harm to mean only an act directed against an individual plant or animal. It removes all of the definitions we've worked with for decades that allow us to explore harm to habitats, habitat degradation, development, mitigation for development. It would remove the need for all of that. For any of you who have worked on development comments, I work on them for THS, I will also work on them for the Desert Tortoise Council. That is devastating. Tucson Herp Society is preparing a letter, which we will send. The letters are due May 19th. Many other groups, some with much more legal expertise than I have, are preparing comments, for this is a legal manipulation based on the dissenting opinion of a former Supreme Court judge who decided that everybody knows that harm only applies to if you whack an individual animal. That was Judge Scalia. So what I'm offering in this email is, if you can in any way prepare your own letter or join with other groups who are preparing letters, I assure you this is just the beginning. The next step is going to be to sunset all of the declared species after 2 to 3 years. And you have to do them over again. And if anyone has tried to work with a species, they didn't have enough staff a decade ago, and they certainly don't now. So the devastation is going to continue, and I guess I would ask you all to care and become involved in whatever way suits you. I know that we all need respite from the stuff that's going on. I have my own. I read history. You read about the Gilded Age, and you know that this is not a unique time, except in the particulars. So please, we have an email. If you have any questions, I'd be happy to try to supply more.
Meaning the Pentagon. And this is what happens when the entire Pentagon is working against you. And unfortunately, there have been people at that building who don't like the change the secretary is trying to. I truly regret agreeing to accept the funeral, Donald. They completely ruined everything I had so carefully prepared.
Can't ignore it.
I spent
So do you think
$20,000
the best thing we can do at this
on
point
nail,
is, right?
and I
Do you
even
think the
prepared
best
black
thing
high
to do at
heels,
this point
despite
for
being 55
individuals
years
is
old
right?
and dealing with
Yes,
varicose veins. I was willing to adhere to traditional attire as a sign of utmost
If you write a
respect.
letter, if I'm
Look
gonna
at
give
what
you a
my
piece
dear
of advice, don't
husband showed
emote.
up in a simple blue
Don't do
suit,
emotion. That's
standing
useless.
out
Your letter will get
conspicuously
toasted.
among the sea of black. Now, I'm not
Look
blaming him
at
for choosing
something
me.
about
That's his
the
fashion
issue
choice.
that
And
is
I have no right
specific
to interfere. What
and
bothers
address
me
that
is the excessive
specifically.
attention it brought.
Preferably
It seemed
from your
reflected
own
sunlight,
abilities. If you
highlighting
have
every
particular
wrinkle on
abilities
my
and
face for all
understandings.
to see. I just couldn't bear it. I mean, I was
We know these
completely
letters are not going to
barefaced,
change the
and
register.
now those
But a
internet
lot of
users
a
are
lot
saying,
of
I
our
look
groups
20
are
years
also
older. Can you
preparing
please
this.
not be
Is
so
there a
harsh?
template letter?
Whatever Mr.
No,
Donald did,
You have to do your
at
own.
least I've always conducted myself with
It's
dignity and
better.
grace. When I met
Short
the Pope for the
letter
first time,
coming from
I wore
you
the most
that is
formal
specific
attire.
and
My
that says,
reverence for
I
him is
know
clear for
that this
heaven
is
to see.
wrong because, not because I love animals or I love plants, but because whatever you can say. So shorter is better than longer, unless you want to do a complex legal thing, which we're leaving to the people who really know how to address this. This is a legally based thing.
I'd be good to maybe a list of talking points or specific things that people can.
Well, it's in the article,
Okay.
but it's in the connections. You can find plenty of talking points. All you have to do is read the front piece to the Endangered Species Act, where the first one says, we realize that habitat destruction. Just pick up on what the act says, on what the register says, and say, no, that's wrong. And here's why I know that that's wrong. All right, thank you.
Thank you for coming to the.
Great. Yeah, I'm not giving. No, this is news. Right. Cleaning stuff out.
Oh, yeah. Party. We sort of used to have a board party in North. It seems harder to get people together like this.
Yeah.
Like anyone else.
The breeding season.
I know you do.
Yeah. Backyard, and they're breeding all summer long. You could do both seasons, like.
Yeah.